So my lab studies mechanisms of body-brain communication. So the brain can exert powerful control over bodily physiology. And when something goes amiss, you have low oxygen in circulation, high blood pressure, not enough nutrients, the body sends signals that control our most fundamental physiology and behavior up to our brain. And Monty Python first realized this, that it's really dangerous to ignore your bodily signals. So I was going to play the video, but I watched it again, and it's actually quite graphic, so I'll just describe the scene. So there's a gentleman who's in a restaurant, and he eats, and he eats, and he eats. There's a lot of emesis going on, and at the end, he's very full, and John Cleese asks him if he would like one wafer-thin mint, and to which he ignores his bodily signals. I'm sure his vagus nerve, the stomach stretch receptors are firing like crazy, but he ignores these signals to disastrous consequences, and he ultimately pops, right? So I guess the moral of the story is don't ignore your bodily signals. And we're just now beginning to understand how these signals how they function at a molecular and cellular level, and how that knowledge can be used to harness new types of physiologies. So one example are the powerful new weight loss drugs based on GLP-1. GLP-1 is a hormone that's part of the gut-brain axis and signals a sense of satiety. And as we learn more about these different types of signals, the sky is the limit for imagining what sorts of new therapies you can develop to control physiology. In recent years, a lot has been uncovered about how our external senses work, our senses of touch, taste, vision, smell, hearing, that enable us to perceive the outside world. And the discoveries of sensory receptors that underlie these systems were foundational achievements in neuroscience. A lot less well understood are internal senses within our body that survey the state of internal organs. And so today I'm going to initially talk about the vagus nerve. It's the 10th cranial nerve and a major body-brain connection that controls basic functions of the respiratory, the cardiovascular, the digestive, and the immune systems. And it has a strikingly beautiful anatomy. So it's a two-way information highway. There are both sensory neurons that take information from the body up to the brain, as well as motor neurons that go in the opposite direction as the principal arm of the parasympathetic nervous system. Today I'm going to focus on the sensory neurons, whose soma live in these what are called ganglia, this collection of cell bodies at the base of the skull. And from there they send projections throughout the body to the heart, the lung, the stomach, the intestine. And these are among the longest neurons in your body. So one end will interface with an internal organ. The other end will cross the skull and target the brainstem, where the neurons activate neural circuits that control sort of these whole body physiological responses and evoke perceptions. Now, the vagus nerve detects a tremendous diversity of stimuli. So in the airways, there are neurons that detect stretch of the lungs with every breath, irritants that make you cough, closure events that make you gasp. In the cardiovascular system, there are neurons that detect stretch of the heart with every heartbeat, or changes in blood pressure, or low oxygen levels that make you hyperventilate. And in the gastrointestinal tract, there are neurons that detect stretch of the stomach, as went awry in the Monty Python case. There are also detection of nutrients that make you feel full, and toxins that make you feel sick. And when we began this work, how the vagus nerve detected anything was unknown at a molecular and cellular level. And these are fundamental problems both in sensory neuroscience and medicine. And also, the number of sensations mediated by the vagus nerve had not been counted. So we have evidence that there are additional body-brain communication pathways that had remained hidden to the field. So over the past, I guess, 16, 17 years now, it's been a while, we've used a variety of approaches to chart the diversity of vagal sensory neurons. Most recently, Sarah Prescott, a postdoc in the lab, used single-cell transcriptomics to chart the diversity of vagal sensory neurons. And this data, what you're looking at here, this is actually from not only the vagal sensory neurons, but also glossopharyngeal sensory neurons in the mouse. The glossopharyngeal nerve is the 9th cranial nerve, and in the mouse, these two ganglia are fused. And what Sarah observes is really a staggering diversity. So a few dozen, I estimate about 37 distinct neuron types using conservative clustering algorithms. And so for comparison, in the visual system, you have rods, cones, melanopsin cells. In the taste system, there are 5 taste cell types for salty, sour, bitter, sweet, and umami. Here we have as much as an order of magnitude greater diversity. And I think it's exciting to think one neuron type might innervate the lungs and control breathing. Another might innervate the stomach and control feeding. Now, there are more neuron types than known reflexes, so it suggests that there are additional body-brain communication pathways that have remained hidden to the field. And so our approach to understand what each of these neurons do has been a genetic one. We've built Cre knock-in mice. So Cre recombinase is a powerful tool in the neurogenetics field. It allows you to sort of tap into the toolkit that neurogeneticists use. So you can use something called optogenetics to activate the neurons. You can ablate the neurons and determine the physiological function of the neurons. You can map their connectivity in the body and in the brain. And you can image their responses in real time to determine what the different neuron types detect, and ultimately try to find sensory transduction mechanisms used by these neurons. So today, I thought I'd focus on the cardiovascular system. We've been interested in all these systems in the lab, but today I'll just show some recent work that we've been working on neurons in the heart and the arteries. So over the past 100 years, since the 1920s, there have been a handful of vagal and glossopharyngeal reflexes mediated by neurons in the arteries. So there's the baroreceptor, which I'll talk about in more detail. There are a couple other reflexes. There's something called the hypoxic ventilatory response, where low blood oxygen, as I mentioned, triggers hyperventilation through a pathway that's not entirely well worked out. There are also neurons that mediate a so-called Bessel-Gerish reflex. This is a defensive reflex that's also poorly understood. The baroreceptor reflex is perhaps the best known reflex. This is a mechanosensory reflex that detects changes in blood pressure and elicits a reflex compensation in real time on a moment-by-moment basis to stabilize cardiac output. And so with every heartbeat, the pressure pulse that emanates from the heart radially distends the arterial walls. And there are sensory neurons that wrap around the arteries and are perfectly poised to detect this arterial distension. And the magnitude of the distension correlates with blood pressure. And so these neurons are stretch receptors in the arteries that then send the signal up to the brain, engaging this parasympathetic and disengaging the sympathetic system to lower blood pressure and heart rate in compensation. And so how the baroreceptor reflex worked has been a mystery over 100 years since it was first discovered. And so we wanted to tackle this question. As a first step, we wanted to identify the neurons that were responsible for the baroreflex. And we turned to an approach called optogenetics. So we expressed channel rhodopsin. This is a light-activated ion channel. We can express this in vagal sensory neurons. First, we express it in all vagal sensory neurons using this VGLU2 driver. And then we can activate the neurons just by shining light on the nerve trunk. And when you activate all vagal sensory neurons using optogenetic approaches, you can powerfully control autonomic physiology with light. You can impact several major physiological systems. So the animals stop breathing, their heart rate and blood pressure plummet, you get changes in gut motility and gastric pressure, and depending on the depth of anesthesia, animals will repetitively swallow and gasp for the duration of the light stimulus. Now, if you do the same experiment, but activate small groups of vagal sensory neurons, now more selective physiologies emerge. So if you activate just vagal P2RY1 neurons, a couple hundred neurons in the entire mouse, the animals stop breathing, but over a 10-second trial, there's no effect on heart rate or gastric pressure. If you do the same experiment, but activate a different group of neurons that contain this orphan GPCR or GPR65, you see an orthogonal phenotype. Now, there's no effect on breathing or heart rate, but you get a reduction in gastric pressure. And so these findings indicated that individual sensory neurons can engage these whole body physiological systems with high selectivity, that there's a division of labor among these different vagal sensory neurons. So which neurons are relevant for the baroreflex? So we've tested a number of different Cre lines to gain access to different small groups of neurons. And we found that activating neurons containing the protein piezo2 phenocopy the baroreflex, and they get this sudden drop in blood pressure and heart rate, while activating other neurons that innervate the arteries have no effect. And so as background, maybe piezo2 doesn't need too much background in this day and age. Piezos were identified by Ardem Patapoutian at Scripps. They're ion channels, mechanically gated ion channels that act in the absence of auxiliary factors. And Ardem showed that they're important for our senses of touch and for limb proprioception, knowing where our limbs are in space. And so we were excited to see that piezos were also expressed in vagal sensory neurons. About a third of the vagal sensory neurons have piezos. And so we wondered whether they might contribute to internal organ mechanosensation as well. And so we reached out to RDEM and began what's really been a fantastic collaboration. Now, if we use genetic tools to ablate the piezo2 neurons, we can attenuate the baroreceptor. So we can induce the baroreceptor in mice by injecting phenylephrine. Phenylephrine is a vasoconstrictor that increases blood pressure. And when you inject phenylephrine in mice, you see this spike in blood pressure. And then secondary to this increase in blood pressure, you see a drop in heart rate through the baroreceptor. Now, if you do the same experiment in animals that lack piezo2 neurons in the vagus nerve and glossopharyngeal nerve, then these animals, when you inject phenylephrine, also have a spike in blood pressure. If anything, this spike is more exaggerated, but the heart rate response is severely attenuated. So piezo2 neurons are required for this baroreceptor. Now, is piezo2 protein itself required? It turns out, much to our chagrin, that knocking out piezo2 alone had no effect. And we looked very carefully over many phenylephrine concentrations, and piezo2 by itself was not required. And I give credit to RDEM. RDEM was the one who found that if you need to knock out both piezo1 and piezo2 to eliminate the baroreceptor, as you can see here. And this is really important, as I'll show, that knockout of piezo2 had absolutely no effect on the baroreceptor, but that allowed us to uncover a different mechanosensory reflex from the heart. And so we became interested in what the neurons in the heart might be doing. It's been a mystery in the field for some time. It's been known that there are vagal sensory neurons that innervate the heart and act as stretch sensors in the heart. And in 1915, Bainbridge proposed that these stretch receptors were detecting venous return and triggered a tachycardia response, or an increase in heart rate. This tachycardia response was called the Bainbridge reflex, but it's been pretty controversial. In humans, the Bainbridge reflex has not been observed. And we also, at least under the conditions we tested, did not observe a Bainbridge reflex in mice as well. And so this raised questions as to what these cardiac mechanoreceptors might be doing. And so our first clue came from a postdoc in the lab, Zikai Liu, who was very interested in the effects of gravity on the cardiovascular system. And so when you stand up, gravity pulls blood to the lower extremities, and there's a decreased filling of the heart as well as a decreased supply of blood to the head and upper extremities. And this is, as I'll show, there's a rapid neuronal compensation for this change in blood pressure. And if you have deficits in this neuronal compensation, this can cause severe problems. You can associate with fainting and dizziness. And in the clinic, this is called orthostatic hypotension, if you are unable to correct for blood pressure changes. And in the clinic, orthostatic hypotension is diagnosed using a tilt table where patients are rotated across postures while blood pressure and heart rate are recorded. And so we adapted the tilt table test for mice. So Zikai adhered an anesthetized mouse to a rotating platform while he was recording blood pressure and heart rate. And what he sees, or what you can see, is when he rotates mice up to an upright posture, there's an immediate drop in blood pressure due to the effects of gravity. And this is rapidly corrected for by a neuronal pathway that I'll discuss what we think that is now. And if you rotate the mouse back down to a prone position, you have the converse occurring. There's a spike in blood pressure that's rapidly corrected for. And you also have a small sustained change in heart rate associated with each of these posture changes. And so we wondered, what are the sensory pathways that the nervous system uses to detect these changes in blood patterning, blood flow? First, Zikai looked at the vestibular system. The vestibular system is known to play a role in posture changes and correcting the visual field, and we obtained knockout mice that lack a functional vestibular system, and the hemodynamic responses on the tilt table were normal in these mice. So then Zikai did transection of the vagus nerve, and when he cut the vagus nerve acutely, now we've completely eliminated the ability of these mice to correct for the posture change. And if we eliminate just the baroreceptors surgically, you have a partial effect. And so there was another pathway separate from the baroreceptors that seemed to play a role. Now the surprise came when he knocked out piezo2 alone. So as I showed, piezo2 knockout mice have a perfectly normal baroreflex. But these mice still had a deficit in this posture response. So they were unable to correct for posture, for the changes in blood pressure that accompany the posture change. And so combining the surgical and genetic knockout approaches suggested that there were piezo2 neurons in the thoracic cavity, presumably in the heart, that were playing a role. And so we wanted to see, are there piezo2 neurons that actually innervate the heart itself? And so Dave Strolick in my lab many years ago developed a genetic approach to map the anatomy of different Cre-defined vagal sensory neurons. What he does is he takes, again, the Cre mice, and he injects an AAV directly into vagal ganglia that contain a Cre-dependent fluorescent reporter so that he can visualize the neurons and he can look at their terminals throughout the body. And when he does this, you can see these beautiful vagal fibers. So this is an entire lobe. So he's looking at all sensory neurons in combination. And this is an entire lobe of a mouse lung, and you can see these beautiful vagal fibers coursing along the major airways in the lung. We think using smaller tools, genetic tools to mark smaller groups of neurons, we think there are at least 10 different types of neurons in the lung and in the upper airways, only some of which we fully understand. So there's more work to do to understand vagal innervation in the airways. This is the aortic arch, the largest blood vessel in the body, and there's another beautiful branch of the vagus nerve called the aortic depressor nerve that innervates right at the peak of the aorta and ramifies in a saddle-like pattern. And we have identified at least three neuron types that innervate the aorta: the baroreceptors, which form aortic claws around the aorta, the hypoxia sensors, and then a third neuron type that we're calling an orphan neuron type. We don't yet understand what that neuron senses and what it mediates. And we also see fibers in the stomach as well as the intestine. These are the GPR65 neurons, actually. This is a cross-section of duodenum. This is the lumen, and these finger-like protrusions are villi, and the GPR65 neurons densely innervate these villi in the intestinal lumen. So next, we wanted to look at the heart, and we performed a similar analysis, and we looked at a whole mount stain of an entire mouse heart. You're looking here at the ventricle. We see dense innervation of both ventricles and both atria of the heart. And as you zoom in, you can see two different terminal types within the heart that are named based on their characteristic morphology. So one is called a flower spray ending, and one is called an end-net ending. And if you do the same experiment with piezo2 Cre mice, these neurons form exclusively end-net endings in the heart. And another Cre line that marks a different ensemble of neurons labels a different subset of end-net endings as well as flower spray endings. So we think there are at least 3 neuron types that innervate the heart, one of which functions as a mechanoreceptor. So what are these neurons doing, and how might their activity be relevant for the tilt table test? And so Zikai then performed electrophysiology experiments on the vagus nerve, and he's recording from the vagus nerve trunk below where the baroreceptors would be relevant for the aficionados. And then at the same time, he recorded cardiac activity through an electrocardiogram. And what he found was that the vagus nerve fired with every heartbeat, and that if you knock out piezo2, this heartbeat-coupled activity of the vagus nerve is lost. And if you link it up to the electrocardiogram, the vagus nerve is firing twice with each heartbeat, and it's probably more easily seen in this figure. So this is many heartbeats from a single mouse, and we think they're firing at atrial systole and ventricular systole, so at the contraction of each of the chambers of the heart. So how does this activity change on the tilt table? So technically, we could not actually perform these electrophysiological recordings while the mouse is rotating. So Zikai asked what hemodynamic changes were occurring on the tilt table and asked whether those hemodynamic changes impacted the nerve responses. And so using a pressure volume loop on the tilt table, Zikai found that every time the mouse is rotated to an upright position, the stroke volume of the heart decreases by 30%. So you have about a 70% of the stroke volume. And so we wondered whether or not stroke volume was an important variable that influenced these nerve responses. And so what Zikai did is he withdrew blood or introduced saline into the vascular system while recording these heartbeat-coupled responses. And what he found is that the intensity of the responses decreased as he withdrew blood, and increased as he infused saline. And so an analogy to the baroreceptors fire with every heartbeat, and the intensity of their firing importance on blood pressure. We think the cardiac mechanoreceptors also fire with every heartbeat, but their intensity forms on a different but often correlated hemodynamic feature, and that is the magnitude of blood volume at the heart when the heart pumps. So what happens when we activate these neurons? We can use optogenetics to activate just the piezo2 neurons in the thoracic cavity, and what we see is a change in blood pressure that responds. This change in blood pressure is not as strong as what we see for the baroreceptors, but it's comparable to what we see on the tilt table. And we also see a small bradycardia, not the tachycardia that you might have imagined for a Bainbridge reflex. Now, we used a frequency of stimulation that matched, we used a closed loop system so that these neurons were optogenetically stimulated exactly with every cardiac cycle, and that's when we saw the strongest response on the optogenetic experiments. So taken together, this makes a lot of sense. So just to summarize everything I've talked about up till now, when the mice are on a tilt table and they rotate to an upright posture, there's a decreased filling of the heart with every heartbeat. This decreased filling of the heart unloads these neurons. It decreases the activity of these neurons, and that results in an increase in blood pressure and an increase in heart rate that helps stabilize the cardiovascular output in real time. And these neurons are presumably firing active bidirectionally, as we saw on the tilt table. And so we wondered whether we could extend this to other models of changes in blood volume. And so Zekai performed another experiment where he decreased central blood volume during traumatic hemorrhage. And so he could induce traumatic blood loss in mice by a transection of the tail in the presence of heparin to prevent clotting. And when he did this, mice lost about 20 to 25% of their blood within 30 minutes. But despite that, the animals were able to sustain blood pressure for the first 30 minutes and also had an increase in heart rate, which presumably maintains cardiac output in the face of a smaller stroke volume. And the mice survived, about half of them died by about 2 hours. Now, if you do the same experiment in mice that lack piezo2 in vagal sensory neurons, these mice bleed out at a similar rate, but they're unable to sustain blood pressure, and they have a catastrophe in heart rate, and they die sooner. So we think these piezo2 neurons are playing a role in a couple of different hypovolemic challenges where the heart doesn't fill as much. So not only when you change the posture, but also during traumatic hemorrhage. And so taken together, we think this provides evidence that the cardiac mechanoreceptors of the vagus nerve do not mediate the Bainbridge reflex. Instead, they mediate a different reflex that we're describing as a blood volume adaptation reflex. And so we were able to reveal this reflex using genetic approaches because in the past, I think it's been really challenging to understand what these cardiac mechanoreceptors do. The cardiovascular system is a closed loop. And so if you make a perturbation in the force at one site, you're going to impact sensors at multiple sites. So really having the genetic tools was essential. The piezo2 neurons innervate the heart. They form these end-net endings in the heart. They fire with every heartbeat, and the intensity of their firing reports on blood volume. If you optogenetically stimulate these neurons, it causes hypotension and bradycardia, and we did not observe the Bainbridge reflex. And if you eliminate these neurons, it causes orthostatic hypotension and compromises the ability to respond to traumatic blood loss. So taken together, all of our approaches, we think there are now at least 6 different types of neurons, vagal and glossopharyngeal neurons, that innervate the heart and the great arteries. There are mechanoreceptors. The mechanoreceptors in the arteries mediate the baroreceptor reflex. And as I showed through a collaboration with Ardem, this reflex requires both piezo1 and piezo2, or either piezo1 and piezo2. There are separate mechanoreceptors in the heart that mediate a reflex we're calling the blood volume adaptation reflex, and these neurons require only piezo2. There are 2 populations of chemoreceptors, the hypoxic ventilatory response, neurons that mediate that response, and another population of neurons that mediate the Bessel-Jarisch reflex. This is a poorly understood reflex, and we don't understand the signaling pathways that underlie this reflex, so there's a lot more work to do. And then there are 2 populations of orphan neurons, and we don't yet even know what reflex they mediate, and let alone how they do it. So again, a lot of work to carry out moving forward. Okay, so that just gives a flavor of some of the experiments we're thinking about on the peripheral side, on the sensory neuron side, I want to talk a little bit about our efforts to understand how the brain receives these inputs to ensure the appropriate physiology ensues. And so, sensory systems in general are charged with the task of detecting very basic stimulus features, photons of light, sound waves, chemicals, forces, and transforming them into complex stimulus representations and perceptions. And different sensory systems have expertise in extracting particular features. So the visual system is really good at determining an object's position, its form, its movement, while the olfactory system, with this enormous receptor repertoire, is wired for recognizing a diversity of chemicals. And classic circuit motifs have been identified that help enable these transformations. So examples include the beautiful work of Richard Axel and Linda Buck, who identified this olfactory receptor-guided spatial map in the olfactory bulb. Another example is work from Wilder Penfield in the somatosensory system. So Penfield, you may be familiar with the cortical homunculus. So Penfield was recording from epileptic patients, again, experiments we can't do today, and applying different tactile stimuli. And he found that there was a map in somatosensory cortex where different regions of the body were represented in different areas of the cortex. And if you, the areas with higher tactile sensitivity have larger domains in the cortex. And if you sort of redraw the human body based on the extent of cortical representation, you get these exaggerated creatures with huge hands and huge lips and a very small body trunk, and that's called the homunculus. And so while there have been major advances in understanding how our external senses work, much less is known about how the interoceptive nervous system, how body-brain signals are organized centrally. And that the interoceptive nervous system has its own unique challenges and presumably requires a distinctive neural circuit architecture. So sensory neurons from the lung that control breathing arrive in the brain together with sensory neurons that control feeding from the stomach. And so how does the brain receive these inputs to make sure the right physiology ensues? And so we wanted to track how the nervous system represents bodily signals at various stages. So we started in the periphery at the level of the ganglion. And this is work from Erica Williams, who is a graduate student in the lab. And she's recording, she's using calcium imaging to measure the responses of neurons in vagal ganglia while the connections to the body are intact. So she uses a protein called GCAMP. This is a genetically encoded calcium indicator, and it provides an optical proxy for neural activity. And so the first thing I'll show you is she just puts an electrical stimulator on the nerve trunk and gives pulses of electricity. And this is what she sees. So this is vagal ganglia, GCAMP fluorescence. She gives pulses of electricity, and with each pulse, she can see neurons firing. She can image over 100 neurons per session. She has single neuron resolution, and then she can genetically tag the neurons using our Cree lines. And then she's moved to other stimuli, so she can stretch the stomach by inflating a surgically implanted balloon, and this activates a minority of vagal sensory neurons. She can then inject liquid diet into the intestine, and this activates other neurons, or she can force air through the trachea to inflate the lungs, and this activates yet other neurons. And so she can apply these stimuli in tandem in the same mouse and see that neurons can have selective response properties. And moreover, neurons are intermingled in the ganglion in a salt and pepper, with a salt and pepper organization. So there's a lack of spatial structure at the level of the ganglion. So what happens as we move from the ganglion up into the brain? Okay, so I mentioned this approach we use to track the anatomy of different types of vagal sensory neurons across organs in the body. We can also see the other end of the organ as it enters, other end of the neuron as it enters the brain. And that's what you're looking at here. So first we're looking at all vagal sensory neurons and how they project into the brain. This is the fiber tract, the collection of axons coming from one ganglion. And then there are dense arborizations in a few brain regions. This is called the nucleus of the solitary tract, or the NTS, which is a major site. There's also a region called the area postrema, which we've also been very interested in, that plays a role in nausea-related behavior here. And then there's a third branch that splits off and heads to the spinal trigeminal nucleus in the spinal cord. And those are mostly neurons, they're jugular neurons that take information from the very upper airways. Now, when you do the same experiment, but you mark small groups of vagal sensory neurons using different Cree lines, now you see a much more refined topography emerge. So here we're activating, or we're visualizing the axons of neurons containing the GLP-1 receptor, or this orphan receptor GPR65, which I showed before innervate intestinal villi. So the GLP-1R happens to mark most of the mechanoreceptors in the gut. And these two neuron populations target immediately adjacent but different brainstem regions. And so there's a remarkable organization that occurs at the level of the axon terminal in the brainstem, and this gave rise to the idea that there might be a map of visceral information directly in the brain. And so a postdoc in my lab, Chen Ran, wanted to test this hypothesis, so he performed a similar series of studies to do calcium imaging directly in the brainstem. And this was a Herculean effort on his part. I'm going to undersell it by a lot. This is a very sensitive area of the brain, and so surgeries are very challenging here. And there's a dense layer of neuropil, or axon bodies, that sort of cover this region of the brain, which causes issues of light scattering and image segmentation in the imaging. And so he used a bunch of technical tricks using, for the aficionados, red-shifted opsins and nuclear localized GCAMPs in order to see signal deep enough in the NTS. And he was able to record responses of these NTS neurons to various stimuli. And so what he observed was that there was indeed this spatial map in the brainstem. So he can stretch the stomach by inflating this balloon, and this activates neurons in a very stereotyped location of the brainstem upon repeated stimulation. And he can use this as an anatomical landmark to map the positions of neurons responsive to other stimuli. So then he can stretch the intestine, and this activates neurons that are nearby, but in a different location. The centroid of the neurons responsive to these stimuli are about 60 microns apart, which is not huge, but it's large enough that Chen could effectively predict where the intestine stretch neurons would reside before he even begins applying the stimulus. And then you can look, for example, he can perfuse water through the larynx, and he activates neurons in a different location. So there seems to be a topographical map that's at least partially linked to the position of the stimulus in the body. And we're at early stages. We need to test a variety of other stimuli as well across these organ systems. But it gives rise to the idea that maybe these are different sensory processing modules that are dedicated to particular stimuli. And we next looked at a variety of Cree-defined cell types in these regions. And what we think is that the organization is a little different than in the peripheral sensory neuron. So one Cree line is reused across these representations. And one representation contains a heterogeneous ensemble of cell types. And so at this stage, we think there's this remarkable topography that emerges, and it emerges from disordered representations in vagal ganglia, at least partially, presumably through this remarkable axonal sorting that we observed. So I want to draw some conceptual analogy to the olfactory system, which has a similarly beautiful, or perhaps more beautiful, map. In the olfactory system, there are over 1,000 odorant receptors and TARs. And each sensory neuron expresses one and only one allele of an odorant receptor, which defines the properties of that sensory neuron. And these neurons containing the same receptor are sort of distributed in a stripe or a zone of the epithelium mixed with other neurons. But then the axons do something remarkable. They coalesce. They come together at single pairs of points in the olfactory bulb called glomeruli. And there are 2,000 glomeruli that dot the surface of the olfactory bulb, and each one is an innervation point for neurons containing one and only one receptor. So through this remarkable organization, if an odorant enters the nose, you'll get this distributed pattern of neural activity in the epithelium that's converted into a highly organized map in the brain. And so we think there are a lot of parallels between the first cranial nerve and the 10th cranial nerve, the vagus nerve. And so similarly, we see that the sensory neuron representations in the periphery are disordered. There's a lack of spatial structure. You have this remarkable axon sorting where neurons conveying different sensory stimuli are sorted based on the information they carry into topological domains within the brainstem. So I think position is really important in understanding how the brainstem conveys these signals to nuclei that control breathing and heart rate, and we're very interested in understanding how that happens. Okay, how am I doing on time? Okay, so I'll try to I'll do my best to get through this one more story. So as another example of physiology that we've been interested in, we've also been interested in understanding the neural basis of sickness behavior. So everyone knows what it feels like to be sick. Sickness is a neuronally orchestrated state that involves many shared symptoms, fever, lethargy, decreases in appetite. You just want to curl up in bed all day. And in addition to general symptoms, there are also symptoms that are tailored to the sites of infection. So a respiratory infection may make you cough, while a gut infection may give you nausea or diarrhea. And so how does the nervous system track the location of the infection to ensure the appropriate physiological response ensues. And so today I'll talk specifically about our work on nausea. So nausea is a signal, as you can see from this poor lady, she's holding her gut. So this is a signal that rises in the gut and is transmitted up to the brain through mechanisms and pathways that we don't well understand. So a variety of stimuli can induce nausea, including pathogens, toxins, drugs that treat cancer, diabetes, obesity, disorienting motion, pregnancy. And it remains a major clinical need because the current drugs only offer partial efficacy. So cancer patients still cite nausea as the main reason they can't adhere to treatment regimens. So I think understanding the molecular basis of how nausea arises is really important. Now, there's a tiny brainstem nucleus called the area postrema that plays an important role in nausea responses. So in the mid-1900s, it was shown that electrical stimulation of this region caused sickness and emesis behavior. Since then, it's actually been poorly studied, but it's a fascinating structure. So it's what's called a sensory circumventricular organ. So what that means is it's in a region of the brain that has a compromised blood-brain barrier. So normally, neurons are protected by this blood-brain barrier, but there are a few sites where the brain can communicate directly with the bloodstream. And the area postrema is one of these sites. So it can literally sample the contents of blood. So in many ways, a sensory structure within the brain itself. And you can visualize this if you inject a neuronal dye, fluoro gold, into the vasculature. This will beautifully label these area postrema neurons. Now, if you remember, I also mentioned that the area postrema was a target for the vagal sensory neurons as well. We don't understand which vagal sensory neurons are communicating to which area postrema neurons. We don't understand the logic of the system yet. And we also don't understand how individual area postrema neurons integrate the responses from the bloodstream with the responses from the vagus nerve. And so we wanted to get a foothold on this system as well. And so Chu Zhang, a postdoc in the lab, also performed single-cell transcriptomics on the area postrema. And here she found a smaller collection of neuron types. There are 4 types of excitatory neurons and 3 types of inhibitory neurons. And she also assembled a genetic toolkit to access each of these neurons. I just highlight a few of our favorite tools. So we built a GLP-1 receptor Cree mice, which actually label 3 of these area postrema neuron types. You might be familiar that the GLP-1 receptor, a major side effect of these semaglutide drugs is that they cause nausea. And we think that the nausea evoked by these drugs involves different ensembles of neurons from the satiety effects, which creates an exciting opportunity potentially for selective circuit control. Another interesting population, which we'll come back to, is this GFRAL. GFRAL is a GDF15 receptor. These neurons are also neurons that they express multiple receptors for nausea-inducing stimuli. So these neurons seem to be particularly critical for nausea responses. And so nausea, it turns out, is very difficult to study in mice. So mice don't display emesis. But they do display behavioral responses to the same poisons that make us sick. And probably the strongest response that is known is that they display a behavioral change called conditioned flavor avoidance. And this is an assay where you have a mouse experience a novel flavor, water containing a novel odor that they haven't experienced before, and you pair that with injection of a nausea-inducing toxin. And then mice, you ask 2 days later, the mice prefer or avoid that odor, solutions containing that odor. And if you have a saline control, if anything, the mice prefer the odor they had experienced. But if you had coupled a variety of different nauseogenic stimuli, the animals will avoid that odor, and that includes the cancer drug cisplatin, lithium chloride, lipopolysaccharide, as well as the GLP-1 receptor agonist xenin-4. So we asked which of these area postrema neurons also can condition flavor avoidance and induce nausea responses. And in the interest of time, I'm running out of time, so I'll just say that these GFRAL neurons are really important for certain nausea responses. And if you activate them, you artificially express a designer receptor in these neurons that they don't normally express. And then you take a ligand for that receptor, that ligand will also condition avoidance to novel stimuli. So, we can also eliminate these neurons and we lose behavioral responses to certain toxins. And so why I'm focused on the GFRAL neurons is that we have some exciting data about the inhibitory neurons in the area postrema. In addition to the excitatory neurons that induce nausea, there are also 3 types of inhibitory neurons in the area postrema. And so what do they do? Well, we know that the excitatory neurons have long-range projections out of the area postrema to a number of autonomic nuclei, the NTS, that I talked about before, sympathetic and parasympathetic nuclei, and a structure, the parabrachial nucleus, where they have subtype-specific patterning. The inhibitory neurons instead project locally within the area postrema. And so this gave rise to the idea that some of them might inhibit the functions of nausea neurons, including those containing GFRAL. And so there were three types of inhibitory neurons. I'm going to talk today about those containing a receptor for another gut hormone called GIP, or glucose insulinotropic peptide. So we obtained Cree mice to access these neurons. And if you administer, in brainstem slices, if you administer GIP, it will activate these neurons, at least the neurons that contain the GIP receptor. And if you record from neurons that lack the GIP receptor, there's no response, although a subset of the neurons were actually inhibited by the GIP receptor. And so we wanted to know which neurons were the GIP receptor neurons we're talking to. And so this is a complicated slide, but through a tour de force, Chu-Chu Zhang in my lab recorded from each of the types of excitatory neurons specifically, in which she used Cree lines to mark the neurons. And she found that the GIP neurons evoked inhibitory currents specifically in the nausea-promoting GFRAL neurons. So this is an inhibitory pathway that was communicating to the nausea neurons locally within the area postrema, and it was a pathway that was responsive to gut hormones. So we wondered, is this an anti-nausea circuit? And so we can give mice GIP, and this GIP prophylactically will suppress nausea responses to GDF15 and to lithium chloride, which also requires the GFRAL neurons to elicit effects. And we can eliminate the area postrema GIP receptor neurons, and this prophylactic response disappears. So just to summarize, I went through it very quickly, so I'm happy to take questions, but I wanted to finish on time. What we think is happening is that various poisons, like GDF15 and lithium chloride, trigger nausea responses to these GFRAL neurons, and that there's a population of inhibitory neurons that responds to a different gut hormone, GIP. GIP will activate these neurons, suppress the activity of the GFRAL neurons, and this leads to nausea suppression. So we're excited when you think about blocking nausea, you could either pharmacologically inhibit the key nauseogenic signals, or you can use activators of the inhibitory neurons as an orthogonal approach. And I should say there's a tremendous interest, obviously, in GLP-1 drugs these days, and combination therapies with GIP and GLP-1 have been tested in the clinic. And Takeda recently observed that GIP suppresses some of the nausea responses in clinical trials. So I think it's really exciting to see this convergence of cellular and mechanistic data with the clinical data that's seen in humans. Okay, so just to summarize, we initiated sort of this analysis of body-brain signaling initially through the vagus nerve. We charted cell types, we generated genetic tools, and we adapted approaches for optogenetics, imaging, mapping of vagal sensory neurons. And today I focused on vagal sensory neurons in the heart. We've also been interested in neurons across the body in the respiratory system and in the digestive system as well. And our ultimate goal is to understand how to identify reflexes and understand how they work at a molecular and cellular level. And I'm very lucky to work with the people that I work with, so I want to make sure I give credit to the fantastic people who did the experiments I talked about today. So Ray Chang was a postdoc in the lab who developed the optogenetics approaches for the vagus nerve. He found a role for piezo2 and airway stretch sensation, which I didn't talk about. He's now a professor at Yale. Sarah Prescott did the single-cell sequencing that I talked about. Sarah also studied neurons in the larynx that initiate airway defense reflexes, and she's now a professor at MIT. Chen Rand developed the imaging in the brainstem, revealing this topographic map of bodily inputs. Chen is now a professor at Scripps. Chu-Chu did the work on nausea-related behavior that I talked about very briefly. I apologize if that was quick. She's now at UCLA leading her lab, and Zikai led the studies that we just published on the mechanoreceptors in heart, describing this vagal reflex to blood volume changes. And Zikai will be looking for jobs soon, so I'm sure he'd love to come to New York. And with that, I'm happy to take any questions.
First question here. Can you also use your microphone?
I've heard so much about this microphone system, so I want to see it in action. I sleep lying down usually. You know, that there is sort of a rotation. The heart will be in the same plane as the body, depending on where, unless you sleep standing up, it won't be in the same plane depending on which way you're rotated. But you know, sleep in the vagus nerve is actually very interesting. So
it does affect. I mean, the sleeping posture does affect.
So it probably won't affect these neurons because it won't change the filling of the heart.
It definitely affects the kidneys, though.
Okay, I can learn. How does it affect the kidneys?
I don't know the technical terms or how to do it, but I've been told, you know, if you have an incontinence issue, it's intensified by sleeping, you know, by laying horizontally. Whereas if you're sitting up, you don't have that much of an issue.
Okay, so so through other pathways separate from the cardiac mechanoreceptor.
In terms of more holistic pathways on stimulating like the parasympathetic nervous system through the vagus nerve, what evidence is there that supports either fitness or hot and cold therapy or deep breathing, things like that? What would you say is the most prominent outcome?
Okay, so therapies. So there's, yeah, that's something we haven't, you know, looked at as much as you know. We play with mice, right? And we're really trying to understand the fundamental mechanisms by which how the neurons fire, what they respond to. So, you know, does yoga work when you're listening to your heart rate and listening? You know, people. I don't do yoga, but you know, there are reports on these sorts of things. So I don't have any anything to add to the sort of the clinical side.
From the back row.
I was told not to drink anything to make sure the mics were okay. So. Yeah, that's $1 million question. I'd love to be able to cure, but I think in the clinic, it's been assumed. So she asked, how could you translate this to curing patients who might have orthostatic hypotension? And I think in the clinic, it's been typically diagnosed that there's, you know, baroreceptor deflex has been the assumption that that's the underlying cause of orthostatic hypotension. So the idea that there's a second pathway where dysfunction could arise, I think is important to understand. How to treat that is, of course, a challenge, right? You know, you need to know what underlies the specific deficit in that patient that leads to the aberrant signaling, right, or the inability to signal. Is it the neurons, the tissue is now fibrotic, so it can't sense stretches efficiently anymore? Are there atherosclerotic plaques that prevent signaling in the baroreceptors? You know, baroreflex sensitivity is a major predictor, for example, of mortality across the board, but it's not really clear how to reverse that. So I think we need to understand the first step towards developing therapies to treat it would be to understand what are the dysfunctions that are occurring that lead to desensitization of these reflexes, and if you can understand how that's happening, then maybe you can come up with ways to treat it.
Just a question. Can you describe how a structural anomaly in the heart, like a right bundle branch blockage, could affect the baroreceptors or that actual regulatory function of the nerves?
So right bundled block. So you mean by something that would cause myocardial ischemia?
Yeah.
So we're working on that. There was a paper that also came out from a lab at UCSD that reported that, you know, myocardial ischemia will triggers a maladaptive response. We had some preliminary data that showed the opposite effect. We need to do more experiments on that. You know, the idea that somehow myocardial ischemia it definitely activates sensory neurons acutely. The mechanism by which that's happening is completely unclear. So we're very interested in that. More to come, hopefully soon.
Thank you.
We have a question behind this pillar. Yep.
Am I on the wrong side? Right. So these are really difficult experiments. So we're increasing the sample size as we go. So we more than one, but so I would say within brainstem you have macroscopic order, but there's mesoscale disorder. So at the interface between these boundaries, things are intermingled quite a bit, if that makes sense. Chen could predict where where the neurons would be. So again, there is this conserved stereotypy across animals. I can't give a quantitative estimate of that at this point until we have increased N, right?
Yes, right here.
So mice aren't people, although they're like people. Do you think all 30 kinds of neurons that you find in mice show up in people?
Yeah, so there are maps being developed for the human vagal ganglia. We have actually my former postdoc, Erica, who's now my former student who's now doing a postdoc in another lab, is working on that. So there will be more to come. I think across sensory systems, there's a lot of conservation. The visual receptors are orthologous, the olfactory receptors are orthologous. We don't have a VNO. Mice don't cough. There are interesting species-specific wrinkles, hiccups, that I think are also interesting, but there have been, there's also a lot of conservation. So time will tell. We'll see.
Question in the back here.
So when you are looking at the NPS, that's also the pathway where the gastrointestinal information passes. So is there like cross information between the nausea pathway and the gastrointestinal?
Yeah. So the taste information is in the very front of the rostral NTS, the front of the NTS, and the visceral information is more caudal. So that's been known for some time that there was sort of this crude division of taste versus visceral, and then we're adding to that beyond taste versus visceral, there's also a more refined map. There is extensive crosstalk within the NTS, and Zach Knight just had a paper where he thinks that taste information, actually not through the rostral NTS, but further downstream, goes top-down to influence visceral responses or has interplay with visceral responses as well. So there's extensive crosstalk at multiple levels. But the initial sensory pathways are segregated spatially in the NTS.
Do any of the neurons that you mentioned today participate in any sort of learning?
So the nausea-related neurons control learning for sure, right? The areopostrema neurons condition flavor points. I showed that with the assay, that there's a novel odor that they then avoid if those neurons are activated. Yvonne de Erjau, who was at Mount Sinai, showed that gut fibers, and Charles Zucker also showed that rewarding signals from the gut to the brain can provide reinforcement signals. So there can be positive signals of learning, and there can be negative signals of learning through these pathways. There's more to do to understand exactly how that happens. But through dopamine release in the striatum is one idea.
Question in the back. Yep.
Do anti-nausea drugs like ondansetron bind to specific populations of cells in the areas you're talking about centrally?
Yeah, so the ondansetron is a blocker of the HTR3A, the serotonin receptor 3A. And HTR3 is not expressed in the areopostrema. It's actually expressed abundantly in vagal afferents. And I've been trying to convince someone for some time to make a flox allele of HTR3A, knock it out in vagal afferents, and see if the ondansetron effects go away. I suspect it has to do with this integration of vagal and areopostrema inputs, vagal and hormonal inputs in the areopostrema. And that somehow there's a gate that certain hormones like GDF15 allow vagal information to flow through in an HTR3A dependent way. But more work needs to be done.
Thank you so much first to Steve for joining us for this presidential lecture.